Okay, while we're waiting for more participants, let's start to give an introduction about this seminar serial. Okay, welcome to join the Statistical AI Methods for the Health Data Science seminars by Startup AI Alliance. Startup AI Alliance is aimed to organize activities that empower statisticians to participate more actively in AI research and leadership, dedicated to enhance the role of statisticians in addressing real-world challenges through AI research, emphasize the dual importance of leveraging both statistical methods and AI tools, ensuring that statisticians are not only participants but also influential leaders in applying those combined approach to solve practical problems. Startup AI has been officially organized as an ASA interest group, and this seminar will be recorded and will be available at the YouTube. And this is the steering committee of the Startup AI and also the inaugural officers. Today, we're honored to have Dr. Olga Vitek as our seminar speaker. Dr. Olga Vitek is a Raymond Bradford Bradstreet Professor, Corey College of Computer Science, and the director of the Barnett Institute for Chemical and Biological Analysis, Northeastern University. She is a fellow of the American Statistical Association and president-elect of the U.S. Human Proteome Organization. She has received many different awards, including the 2021 Gilbert S. Ullman Computational Proteomics Award, Indigo Bioautomation Female in Mass Spectrometry Distinguished Contribution Award, NSF Career Award, and Essential Open Source Software Award of the Chan Zuckerberg Foundation. His main research focus on the use of statistical and machine learning methods to integrate mass spectrometry, proteomics, and system biology. Okay, please join me to welcome Dr. Vitek. Yep.
Thank you so much. Thank you very much for inviting me. It is always a special pleasure for me to talk to statisticians and statistics community. So indeed, as you heard, I have many hats. So currently I'm a professor of computer sciences at Northeastern University. I direct a Institute for Chemical and Biological Analysis, but in my heart, I am a statistician. I was trained as a statistician at Purdue in Indiana, and then I spent quite some years as a faculty in the Department of Statistics at Purdue. And so my training in statistics really inspires and informs a lot of work that we currently do. And so mass spectrometry has a lot of uses, and it can be used to answer a variety of questions. It can be done in a variety of ways. And so I do not have time, obviously, to tell you all about all possible uses. And so I decided to talk specifically about one flavor of mass spectrometry, which has really interesting computational and AI challenges, and this is spatial mass spectrometry. So sometimes it is referred to as mass spectrometry imaging because you can also think about the measurements as producing images, but it is very different from the natural images or, optical images that we can take. And so this is what it looks like. For example, here we have a tissue slice. So this is a mouse brain. And so what we can do is from different locations in the tissue, we will collect mass spectra. So here is a green location, we will collect a mass spectrum. So what mass spectrum is doing, it is taking all the molecules collected from this particular location in the brain, and it separates them. First, it ionizes them, meaning it attaches a small charge molecule. And then it separates them according to their ratio of mass divided by charge. So essentially, heavier molecules kind of fall on this part of the spectrum, lighter molecules fall at this type of spectrum. And so a peak in the spectrum means that the instrument found some biological material at this particular ratio of mass divided by charge. And so then what we need to do is to actually figure out what this is, what kind of molecule, how much of this molecule, how is this molecule distributed spatially, how is it changing between different locations in the brain, how it changes between conditions, and all of that. And so from the spatial perspective, for example, now we have another location in orange, and we have another mass spectrum. And then we take another location in red, and we take another mass spectrum. And so the intensity of the peaks tells us about how much of this molecule is present. And so we can kind of look at this from multiple perspectives. And so one is in the spectral domain. So if we look at one spectrum at a time, there are quite a few computational problems, which can be, signal processing or inferential questions, is such as, well, sometimes different molecules have very similar mass, so their peaks are very close to each other. So how can we separate the abundances of each of the molecules? Then we need, so sometimes it's the opposite problem. The same molecule gives us multiple peaks. For example, there's multiple charge, or maybe some molecules, they break during the process. And so how can we find multiple peaks in the spectra that actually come from the same analyte without necessarily knowing what it is? Then we need to figure out what the analytes actually are. And then this actually is a fairly simple version of that. So there are more complicated versions of that that would, instead of one-dimensional spectra, produce two-dimensional spectra and so on. And so potentially we may want to integrate this with other types of experiments and other types of modalities. So this is in the spectral domain. But then what we can also do, we can focus on a particular ratio of mass divided by charge and essentially take a cross-section of this data structure. And then at each location in the tissue, we essentially have the value of intensity. And so this is what this would look like. And this is what we sometimes refer to as mass spectrometry images, because they essentially tell us about the chemical heterogeneity or chemical kind of composition of or distribution, I guess, of this molecule throughout the tissue. And so here in the spatial domain, we can kind of speak in a more statistical language. And sometimes it is helpful to speak in terms of classes. So things of kind of with some common properties. So for example, class discovery will define subregions of the tissue which have homogeneous chemical composition. For example, with respect to biology, morphology, or disease, or drug being present, or maybe just noise and background. And so class discovery essentially is an unsupervised kind of approach, right, which would look for clusters, right, or similar type of locations in the tissue. Class prediction, so now this is a supervised approach where we want to classify healthy versus disease. For example, one very common application of that is in pathology. So for example, during surgery, it is important to remove the tumor entirely and make sure that we have margins around the tumor that do not contain the tumor, but at the same time, we want to remove as little of the healthy tissue as possible. And so essentially, it's a classification problem. For every tissue, is it tumor or is it healthy? And so traditionally, what people do, they just pour some liquid and they look at the color intensity, but we can do better. We can actually characterize the chemical composition of tumor versus non-tumor subregions and develop a classifier. So this would be the class prediction problem. And then class comparison. So what if we have many of these tissues, some of them have a disease, others don't. So we want to know what is changing in abundance between these conditions, knowing that we have some spatial heterogeneity. And so this is really the extent of questions that we may want to ask by thinking about the spatial domain. And of course, a really important part of this is how do we actually develop tools that allow people to put these methods in practice. So a very important aspect of these experiments is that they generate very large files. So we're now talking over 100 gigabyte per tissue with modern instruments. And you can imagine that we have multiple tissues and so on. So we need to make sure that the methods are scalable. The users of these tools are scientists with very limited computational background. So how can we make this possible for them? And of course, we want to make sure to supports research that is not just accurate and effective, but also reproducible. And this really requires open access to tools and open access to data. And so this is all the issues that we have in the space. And so before I kind of continue, I would really love to acknowledge the members of my lab who work on these problems. And so they are the ones who actually do the hard work, and they are the ones that I want to celebrate. So what I will do is talk, kind of have two parts, and we will see how much time each part takes. And so the first part will be talking about how we approach this from the perspective that is fairly traditional for mass spectrometry imaging. And then the second part would be the perspective that is fairly traditional for statisticians, but maybe less traditional for mass spectrometry practitioners. So speaking about open source tools and making tools accessible to non-statisticians and non-computational folks, a really big effort in our lab is in developing open source software. And so for the last over 15 years, we have been developing Cardinal. And Cardinal is, so Kylie is the lead developer, is a open source R and bioconductor software ecosystem by now, which does a lot of things. So it would import the raw data as they come off the instruments. It can visualize the data. It can prepare the data in terms of finding regions of interest, subsetting, filtering, etc. Pre-processing is a really big part of this, is finding the peaks, smoothing the peaks, calibrating. There's a lot of technical issues while working with very large data. And then, of course, where statistics and AI shine is the next step, which takes this process data as input and hopefully provide some insights. And then we want to export these results. And so this is actually a very comprehensive software system, which I just want to kind of mention, started at Purdue. When Kylie and I were at Purdue, it also continued at Stanford, where we were on sabbatical. And so during this time, we established quite a bit of collaborations with people who generate data for that. And because the data are very large, so this is R in bioconductor, at least as far as the user is facing. But in the backend, increasingly, this is C++ efficient code, in particular out of memory computation, because the data do not fit in memory, parallel CPU computation, and so on. So there is a very non-trivial software engineering that happens behind that. And so I will mention a little bit on pre-processing, but I would really like to focus on statistics and AI because that's what's interesting. And so talking about things that are traditional in the mass spectrometry community. So remember I was saying in the spatial domain, so class discovery, class comparison, class prediction. So class discovery was the first approach that people took to work with this data. And so class discovery essentially means clustering, right? And so here is one example. So this is a pig fetus data set acquired by Graham Cooks at Purdue in his lab. And so when we think clustering, so what can we do? Well, k-means, right? So this will be a very natural way, or maybe doing some kind of PCA and k-means or something like that. And so then if we would essentially look at, the type of segmentation that we can obtain with k-means, either in the original space or in with PCA. So what we notice is that, well, on one hand, it's kind of nice because now each location here, maybe if I go back to this, actually here, each location here is viewed as a list of peaks, right? So it's described as a multivariate vector of measurements. What these methods don't do is they don't recognize that there's a spatial relationship between all these individual locations. And so this kind of segments become very jagged, right? And they probably do not reflect the biology, right? And they also don't tell us which peaks actually are responsible for the segments and which peaks are kind of just go along with the ride, in the ride. And so what the very first thing that we propose to improve on that is actually acknowledge the fact that we have spatially related locations. And so the way we did this was you may be familiar with nearest Shrunken centroids. So this is another version of unsupervised classification, which also incorporates regularization, so that if you have a multivariate vector describing each data point, you can essentially use regularization to eliminate some of the dimensions in the vector that do not contribute to the grouping. And so this is what this looks like. So first of all, we see that now this segmentation is much more smooth. And also, if now this is our vector of peaks that describes each location, so we see that in pink are the peaks or the molecules which actually are responsible for these locations being, for example, the brain. This is the brain of the fetus. And we can also report this as signal-to-noise ratios. And so we see now that parts of the spectra, they're really not relevant for the brain. And then others, high signal-to-noise means that the molecule is enriched, and the low means that the molecule is depleted. And so this is useful because it gives us more smooth segmentation, and it also tells us which molecules are most responsible for all these locations going together in a segment and in which manner. So then the problem with this, though, is that it is a single segmentation based on all the molecules that we have measured. And so what it assumes implicitly that all the molecules have to go together in some segments. The reality, though, is very different, that different molecules can have very different spatial distribution. And so there's really no reason to think that all the molecules cluster somehow in one way in the brain and another way in the liver and another way in the heart. And so what may be more interesting is to think about the segmentation problem, not from the multivariate perspective, but from the univariate perspective. Essentially take one particular molecule and say, can I find segments for this molecule? And then take another molecule and say, can I have some other segments for this other molecule? And this is very useful biologically because different molecules are distributed in different ways. And so this is Dan, who used to be a PhD student in our lab, who graduated. This is what she came up with. And so this is another collaboration with Graham Cooks. And so this one is actually really interesting because unlike the previous one, which is a tissue, this is a oil painting. So this is an actual painting on canvas with oil colors. And different colors have different chemical composition. And so we can actually analyze this with a mass spectrometer and see which colors have different molecular distributions. And as statisticians, we love it because, well, we know the ground truth. We know what we expect to see. And this is actually an Indiana farmhouse. And so we hope that by collecting the data and applying some kind of segmentation, we can reconstruct that. And so this is what happens if we reconstruct this multivariate way. So for example, here, the green color somehow didn't come out. It got lost. The black borders get lost, and the donkey here in the corner also got lost. And so the question is, can we, instead of doing it in a multivariate manner, can we do it in a univariate manner? And so, for example, if we take one particular molecule, so mass overcharge 650, so this is what the image looks like, right? So again, this is just a single image across all the locations. And so now what we can do, we can actually segment this image to have the black color stand out. Or we take another M over Z and we can segment this image in this univariate M over Z way, and we can have the green color stand out. And so the technology there is the spatial Dirichlet Gaussian mixture models. So now this is interesting because essentially here different colors, they do a very different job as we would expect. And so the same thing biologically, you would expect that different molecules can potentially have very different spatial information. Now, the problem with this method is that if we have just one peak, well, one peak can be noisy, right? It can have missing values, it can have outliers, and of course, this will compromise the accuracy of the segmentation. So ideally, if we have multiple peaks that belong together, for example, because they are fragments of the same molecule, we would want to aggregate that. We don't want to do it over and over for each piece of the molecule. The problem is that we do not know which peaks go together, which peaks came from the same molecule. And so this is the work by Dan that tries to improve the property of the segmentation in this direction. And this is done as follows. So here's a simulation where we have a ground truth. So this is a background image. Below is the blue circle, and on top is a gray circle. So the gray circle is slightly smaller, and the blue circle is slightly larger. And so ideally, we would have a cluster of molecules which are gray circle, a cluster which are blue circle, and a cluster that is orange. And so from the simulation, because, well, we know how we simulated the data, we can think of how much the fact that multiple peaks come together for segmentation, how much it helps. And so essentially, if we have only peaks from cluster 2 or only peaks from cluster 1, we do less well than if we find multiple peaks which do belong to cluster 2. And so that kind of inspired this type of work. And so now this is another example, another tissue. This is the optical image. So now here we have a multivariate segmentation. This is what would happen if we use all the peaks and segment the tissue in the space of all the peaks together. And we take one particular ion and we see that this ion doesn't have quite the same spatial information as all the ions, but it is also noisy. And so now if we manage to find multiple peaks that all have the same spatial distribution and then segment based on those, we have a lot more kind of accurate information. And so the technology here involves neural networks because it is a more complex iterative optimization, and the training for the neural network was self-supervised to first bring together peaks that obviously belong together and then little by little add additional peaks as warranted. So this is for the unsupervised methods. So now another aspect is supervised class prediction. So as I mentioned, for example, in surgery, it is very useful to have a training set, which maybe a pathologist will label as tumor versus healthy, and then develop a classifier. And a traditional way, I will not even show the results. You can imagine it can be partial least squares, logistic regression, random forest, neural networks, you name it, right? So pretty much any classifier. So there's a problem with that. And the problem is as follows. So the problem is in the training data. So to train any classifier that we want, we need to have tissues that are annotated previously as, let's say, tumor versus non-tumor. And it has to be done at enough resolution. So the problem is that in practice, it is done by pathologists. And the pathologists, they don't have the same spatial resolution as a mass spectrometer. So what pathologists will do, they will say, okay, here is a healthy biopsy. We know it's healthy because it was taken, from a tissue which is likely not affected. But here is a tumor biopsy, and the pathologist will just draw a circle, and they will say, well, there is tumor in there. But they will not be able to, like, literally pixel by pixel annotate each location in the tissue. So what happens then? So if we now look at the images, so our training set is actually compromised. So maybe our healthy tissue is healthy as we expect, but what we think is a tumor tissue is actually a mix of healthy and tumor. And because our training set is compromised, well, the classifier will attempt to label everything as tumor, and it will not be right. So what we really want, we want to label each individual pixel as either tumor or healthy, so that we can give more accurate representation of the tumor, for example, for the surgery. And so this is what we want, right? We want to label some of the locations as tumor and then the other locations as healthy. And so this is one of the methods also developed by Dan as part of her PhD. So essentially, this is semi-supervised multiple instance learning, which says, well, we can trust that this is normal tissue. Now let's find pixels in this tissue which look like normal, and we will call them normal. And then we will find other pixels which are different from normal, and we will call them tumor. And so this is how we can kind of learn iteratively to classify tumor versus non-tumor. And so we were very fortunate because we had collaborators who actually, pathology collaborators who actually took the trouble to label pixel by pixel every location in a fairly large cohort just for the purpose of helping us understand the classification. And we could show that it really makes a big difference in practice, especially depending on how precise the pathologists are when they label the tissues. So now another aspect which is kind of a little bit adjacent to that, but very important, is to recognize that mass spectrometry imaging is not the only technology that can give us spatial information. There are many others. First of all, mass spectrometry can have many different flavors, and they have different pros and cons. H&E, this is the typical staining-based technology where people look at the intensity of this color to understand what are the different morphological regions. But then also there are transcriptomics experiments and many other kind of hybridization-based experiments. And all of these experiments, they're meaningful and useful because they measure different parts of the molecular space. The problem is that many of them require different sample manipulation. And so the samples become distorted or otherwise affected between different experiments, which means that we can no longer overlay these images on top of each other for joint analysis. And so part of the work in the lab led by Sai has developed coregistration technologies that allow us to overlay multiple tissues in ways they're not just affine transformations, but they're, stretching and compression and fold of the sample on itself, and how we can overlay this across multiple tissues. And so this is also a optimization that involves multi-head attention neural networks. And a big problem here is to understand how we actually verify the accuracy of this coregistration. And so Sai also came up with some numeric metrics that we can work with. And I am happy to tell you more about that. But in the interest of time, so what I would like to do now is to tell you more about approaches which are more traditional and more maybe natural for statisticians, but they may be less natural is in this field yet, in part because of all the challenges of data preparation and data management. And this is class comparison. So essentially comparing the abundance of a particular molecule or many molecules, not just spatially within one tissue, but also across multiple tissues, maybe healthy and disease, or maybe more complex designs. And so I will have two examples, and both of these examples are led by Ethan, a PhD student in the lab, just to show you what the issues are and how our statistical training can help in this field. So the first example is osteoarthritis. So a case study of osteoarthritis, so it degrades the bone and cartilage. And so the statistical question here is, well, which analytes change in abundance between different types of conditions in the knee? And so the conditions we have is medial versus lateral. So for each subject, we have two sides of the same knee, medial and lateral. And then we have subjects with osteoarthritis, and then we had controls, so subjects which did not have any disease. And so those were post-mortem, so subjects who donated essentially their tissues after they diseased. And so this is a fairly complex design by the standards of mass spectrometry imaging communities. So we have 4 subjects, 4 from disease, 4 subjects from control, and then we have a paired design where we have medial and lateral tissues. And what is also interesting is within each tissue, we have part of this is bone and part of this is cartilage, right? So bone is one thing, and then cartilage is what prevents the bones from friction. And so osteoarthritis essentially degrades the cartilage and therefore degrades the bone. And because there is degradation, we expect that there is a change in molecular composition. And so this was actually one of the first experiments with the bone specifically. So technically, it was very difficult to do. And this was reflected in the quality of the data. So this is the images of one particular molecule across all of those tissues. So all of the 16 images. So now if we think of this workflow, as I showed earlier within Cardinal, we can translate this specifically for this tissue. So how do we go about this from the beginning to the end? Well, of course, things that are very familiar to statisticians would be experimental design, sample size calculation, the hypothesis, all of that. So this is very important. But once the data come in, there is a lot of processing that needs to be done. And so the processing, I'll just kind of show very briefly, reading the data and picking. So this is what this looks like. So this is a mean spectrum of a single diseased data set. So this is M over Z. This is the mean intensity. And so this is what the mean spectrum looks like across all the locations of that particular tissue. And so then quite a few steps had to be taken. So pick, normalize, baseline remove, etc. And so this is what our multivariate kind of vector that describes each location looks like after all of this processing. And this is all I will say about that, but just to kind of make sure we know that this has major implications for the quality of the data and the assumptions we make and so on. And so the second interesting thing is to define regions of interest, right? So as I mentioned, we are interested in cartilage. And so what people usually do, and this is also something where as statisticians, we need to change the culture a little bit. People can do segmentation, right? Just using the methods I described, use multivariate segmentation to say, oh, look, this is one segment, this is another segment. So now let's compare, for example, cartilage to the bone, right? As defined by the data that we just collected. And so the problem here, it is double dipping, right? That we use the same data first to define the segments and then to test the segments. So this is a bit of a cartoon if we just have one sample and we will say, let's say low values of this analyte is one region of interest, high values of this analyte is another region of interest. Now let's see if these two regions are differentially abundant. Of course, we will have a small p-value, but this, of course, will be overfitting because we just use the data to define the regions. And the same problem may be a little bit less dramatic, but will still be preserved if we have a multivariate segmentation. So for our problem, this is more relevant potentially, is when we will have two samples and we will use the same data to define what we compare. And so now what we can say, okay, let's say the high values of analytes will be one region of interest and low values will be another. And so we may just define what we compare based on high values of analytes, but this is not really comparable because these are not the regions of the tissue we're interested in. And then we will say, of course, there's no difference because this is high intensity and this is high intensity. And so again, this is now a selection bias where in different samples we choose to select something based on the response variable. And so this is something that actually takes some work to convince our experimental collaborators to avoid this type of data reuse and instead use external markers to define the regions of interest and then eliminate those and make comparisons among all the remaining analytes. And so this is a simulation that illustrates this a little bit more in the image. So we have two conditions, condition A and condition B. So we have this donut that represents the region of interest, and some of the peaks are noisy and some of the peaks are differential. So they change in abundance between condition A and B within the donut. So if I just segment my image based on the features which define the donut, right, define the ROI, of course, my segmentation will be perfect. But if I use some kind of multivariate data-driven segmentation, the noise interferes and my segments are not quite as they should be. And so no surprise that we will no longer be able to detect changes in abundance between conditions. And so it manifests itself from this simulation that the features that define segments in condition A and B are not the same. They do not overlap with true differentially abundant features. And so this is what the differential abundance would look like if we used some external informative marker. And this is what the selection bias looks like. So we don't find any changes because we just use the same data twice. So this is something that is, I hear a lot of discussions along the same lines in spatial transcriptomics as well. But this is something that, as statisticians, our job to remind people of these issues. So back to the osteoarthritis data set. So what we did there, we actually found some markers for background, bone and cartilage, did this kind of iterative, like three-step segmentation, discarded these markers, and then proceeded with tests for differential abundance. Okay, and so, but before that, we also needed to do additional data processing. So this is also kind of illustrating the issues. We had 45,000 raw features it went down to 894 picked features, then removed features which are not informative essentially, and they don't have any signal anywhere. And after that, we also looked for peaks which have presumably come from the same source based on some information and so on. And so, long story short, from 45,000 spectral features, we ended up with 124 features that were used for differential analysis. And this is a big loss of information, and this is why using complementary modalities is very valuable in this field. And so now statistical modeling. So this is the part that probably I don't need to explain very much. So we essentially have a paired design. So two groups, medial and lateral. Within each group, we have a paired design. Sorry, two groups, osteoarthritis and healthy, and then paired design for medial and lateral. And so we can ask questions such as, is there a difference between healthy and osteoarthritis for medial? Is there a difference between healthy and osteoarthritis for lateral? Is there a difference of differences, so interaction? In this case, we didn't find any differences because the signal was so subtle and there was enough noise because remember, bone is very hard to image by this technology. However, this data set is not, experiment is not lost because what we can now do, we can actually do sample size calculation based on that. And we can project how many replicates we would need if we wanted to follow up on this type of study, but with a better powered experiment. And so we can talk about sample size within versus between subjects. If we look for multiple testing, what type of FDR correction we would need. And so we actually can use this data to plan further studies and already make informed decisions about how to do it better and really detect changes in abundance. So this is very useful, regardless of the fact that we did not find the differences. And by the way, this is also something that our experimental collaborators don't always appreciate, right? That there is use of the experiments that didn't find anything conclusively. And so now is another example. This will be my last case study. So here, what we essentially did when we did differential analysis, we said, okay, we found the cartilage, we found the peaks, which we're interested in. Now let's average all the information in this region. And so this linear mixed effects model, they compared average abundance of the cartilage across all the different individuals. Now, sometimes averaging a region of interest is not really what you're looking for when you are doing spatial analysis. Sometimes you really want to understand how the molecular abundance changes spatially without averaging a region of interest. And so this is an example of this type of study. So this was a study by our colleagues which were interested in the distribution of lipids in the brain after stroke. And so this is a mouse study, and they induced, using a surgery, a stroke on the left side of the brain. And so they had 1, 2, 3, 4, 5 mice, and they had looked at the brains or imaged the brains 7 days after the stroke was introduced, and then 28 days after the stroke was introduced. So this is what the experimental design looked like, and they were interested in a particular lipid. So unlike the previous study where we were just interested in general in chemical heterogeneity of the tissues, here they had a very specific lipid, which is known to be associated with stroke recovery. This lipid has a very specific mass, so we can actually interrogate the spectra for this very specific peak. So in terms of spectral processing, this is actually much easier because we have a very concrete question that we want to ask. And so this is where the stroke was introduced for each of the mice. But the question was not really about the region associated with the stroke, but it was associated with these adjacent cortex regions. So brains have two cortices, right? So left cortex and right cortex. One is closer to the region of the stroke, and the other is further away. And also the cortices in the brain, they are not homogeneous, they're structured in layers. And so this kind of attempts to represent the layers. And so essentially, one way to frame the statistical question is the gradient of penetration of this particular lipid throughout the brain, right, through the cortex layers, differs between the left hemisphere, which was near the stroke, and the right hemisphere, which was further away from the stroke. And so we really want to understand not just the average change, but we want to understand the spatial gradient. And so therefore, averaging these regions will not be really useful. So what we can do? Well, we can start with the traditional method. So segmentation is really very widely adopted in spatial experiments in general. And so what we can do, we can apply some of these clustering methods I talked about earlier. And so, well, those methods require us to specify the clusters. Clearly, the more clusters, the larger number of clusters we specify, the more heterogeneous the clusters are. We are interested in these regions of the brain, right? And so we need to kind of focus on this part specifically. So that's what we do. So if we take this, so we essentially segmented out these regions of the brain that we are interested in. Okay, so is it useful? And well, this is again an illustration of selection bias that I mentioned earlier. So if we look at how these segments are distributed through different cortices of different mice, so we see that they're very different. For example, the purple cluster is on the left side here, but then it kind of goes on the right side here, and here it's just in the corner. So the segments, they just capture regions where the abundance of this lipid or multiple lipids is very similar. And so now if we look at the distribution, so the colors here are the clusters. So we have the green, purple, gray, and orange. And neighboring dots, they are from two conditions, 7 versus 28 recovery days. And so we see that there's a lot of difference between the different segments. Well, because that's how the segmentation works, right? We look for subregions of different chemical composition. But if we compare 7 versus 28 days, there is no difference whatsoever. So essentially, segmentation is not helping here, right? It tells us, okay, well, here are the regions which are preserved, but our question is there a spatial gradient? Okay, so then what we can do, we can also take those cortices segments and we can average them. And as I mentioned earlier, this is not very helpful because it doesn't answer the original question. But also if we do that, well, and if we do a test of like just a t-test between 7 and 28 days, we don't really find very much because we averaged out all the interesting information. So this is also not working. So we really need some type of different models. And so this is how we think about that. So now we have some for one particular lipid, right? So we're talking about one molecule. We can think of this as being spatially distributed. So this is the observed intensity of the tissue. So the y-axis is the intensity, and these are x and y coordinates. We can think of this as some kind of surface, right? Or we can also think of this as a heat map representing the intensity of this lipid. And so we would like to model this in a simple case as some kind of smooth surface that we can model and then random non-systematic residual error. So this is what we would do if we had a simple experimental design, but our experimental design is fairly complex, right? It's also paired. We have left and right cortices, two conditions. So now we can imagine we can have two conditions, and essentially our question would be, is the gradient of the surface different between conditions on average over multiple mice and different types of cortices? How can we do it? Well, so now we can actually fit a generalized additive model. So this is just one mouse brain. So this is the outer and inner cortex of the left side, and this is the right side. And we decompose this into the systematic part, the intercept, a similar part of the penetration along the distance. And this is the interaction, how the penetration differs per hemisphere and per recovery days. And then we have the random field that explains just the morphology of the brain and then the non-systematic error. And so we can model this with different types of, approximation, right? Approximating this in a flexible way. And so this interaction is essentially of our interest, right? So is this type of surface different across different hemisphere and day combinations? And so now we can essentially query this model and have model-based summaries. And so now we can look at 7 days versus 28 days. This is the penetration distance. So this is for 7 days. This is the estimated gradient for 7 days. This is the estimated gradient in 28 days. Looks like they're not the same. So we can literally overimpose them and see how they differ. And we can literally take the difference between them, and we can have some estimates of uncertainty. And so now we see that the abundance and the outer cortex is greater at 28 days, but it is greater at 7 days. So this already tells us gives us some information about how much this particular lipid penetrates throughout the cortex as function of recovery after the stroke. Well, so this, I hope, gives you a sense of the different types of methods that can be used in this field. And if you would like to hear more or in general know more about our work, I would really like to share with you this. So for the last 12 years, on the campus of Northeastern University here in Boston, we have organized a 2-week educational program that we call Computation and Statistics on Mass Spectrometry and Proteomics. And in brief, we call it May Institute, May because it happens every year in the month of May. And it is a 2-week program that dedicates a lot of time talking about all aspects of computation, statistics and computation for mass spectrometry. So I talked about imaging, but there is also proteomics, and there are also aspects of tools that interpret the data, R and Python computing, knowledge bases of biological knowledge that can be used to interpret the results. And so even though it is a 2-week program, it is actually broken into bits and pieces for like a couple days at a time, and you can pick and choose which part you want to attend. And we work very hard, and especially this year, we had 3 sponsors that allowed us to waive tuition for most students and postdocs so that they can attend this for free. It isn't person though, so we need to all come together, but it is very nice because in addition to all the scientific information, it is also building a very nice community of people interested in similar topics. And we have people from academia, people from industry. You can really compare notes and compare experiences with many people. So please don't hesitate to reach out. And in fact, you can even sign up for the mailing list if you're interested to know more. We expect to open the registration around December this year. And that's all I had to share. So I just want to make sure I thank the members of my lab who did most of the work. Kylie is the leader of that, especially for all the kind of backends and high performance computing associated with mass spectrometry imaging. Melanie, Dan, Sai, Ethan have led many projects that I just talked about. And of course, we have a number of collaborators who have shared their data and expertise with us. And so thank you very much. And I hope that we have time. I'm happy to answer questions and tell you more if we have a minute.
Yeah, I think we have a question in chat.
Mhm.
Could you please comment on what disease or conditions MSI is most suitable for? And in terms of statistical analysis, then what are the similarity and difference between analyzed MSI data and the spatial transcriptomics data?
Okay, so these are 2 different questions, right? So which disease and conditions it is most suitable for? Well, so I think it is kind of important to think about what kind of molecules can be characterized this way, right? And so this is typically small molecules, lipids, peptides, so certainly not entire proteins. The entire proteins are too heavy. Uh, and so if the disease has an important metabolic component, right, or small molecule component, I think, or lipids are really, really interesting. So I think that would be the best technology. And then the second aspect is the disease that has some spatial heterogeneity. And also, maybe the tissues are not too fatty. So, for example, breast cancer samples are very difficult to image because they don't have enough spatial heterogeneity and they have a lot of fatty content. So it really depends, right? But usually, so this is one. Then another very common application is drug delivery. So if you have some particular compounds and you want to know, does this compound make its way into a particular region of the brain? Well, the last example would be perfect for that, right? So you would think that this compound should reach, a particular brain region. So this would be to confirm that it does, right? So that would be um, another important use case. Now, mass spectrometry versus spatial transcriptomics. Well, when it all said and done, and your data nicely arranged in some kind of array, right, and everything is normalized and summarized appropriately, I think many methods will be very similar. The difference is when you start, right? So the raw data in mass spectrometry look very different from transcriptomics because they are not counts. They depend on ionization, like ion competition, ion efficiency. There's like all these issues that require essentially very specialized signal processing, mass calibration. There's like a lot of very specialized baseline removal, very specialized data processing that is not the same as for transcriptomics. And add to this very large data files. So altogether, this really requires very domain-specific expertise. What we hope, though, is that with Cardinal, we automate a lot of that. So we still need people to kind of understand, what the parameters are, what the methods are, how to work with the tool, but you do not need to reinvent the wheel. You don't need to invent all of this from scratch. And we do export data in a format that can potentially be compatible with transcriptomics. So the problem now, if you want to do a joint analysis, so then not every mass spectrometry and transcriptometry modalities can be run on the same tissue. So sometimes you actually need to have consecutive tissues, meaning they're not the same, right? And so then you need to co-register them, make sure that the correspondence between individual locations in the tissue is the one that you have in mind so that you can really model this jointly. So there is that extra complication. So I hope that that answers. So, but at some point it becomes similar, but it's so much has to happen before the problems become similar that by then, like you solved 90% of the issues already.
Okay.
What are the, yeah, you
Okay.
go.
The next question is, could you please comment on what are the difference between the biological conclusions drawn from MSI segmentation and WSI segmentations in cancer, and what can be uh, disclosed by the discrepancy?
So what's WSI?
Maybe, yeah, can you just unmute
Oh,
your mic?
sorry, it's just the
Yeah.
whole slide
Uh-huh.
image. Yeah, it's just the pathology, yeah. Whole
Oh
slide image.
yeah. Oh, I see. I see. I see. Well, a lot potentially, right? Because mass spectrometry imaging really thinks of kind of characterizes the chemistry, right? The chemical composition. And the whole slide, and especially if it's a HNE. So HNE, what is it? It's just a hybridization, right, with some particular target. So it is a lot more rich than HNE and whole slide imaging. So there's a lot more information, and it can not only tell you this is this subregion, this is that subregion, but it can even potentially give you the information about the mechanisms, what type of pathways are activated, so there's a lot more information there because you really get down to the individual biomolecules as opposed to just staining. But then you need to solve all these technical issues. But I think that this is very complementary. And also there are new technologies, mass spectrometry-based technologies that are developed, which can allow us to see even more molecules and at more lower abundance. So I think that this field is developing very, very quickly. And I really see a lot of applications in the future of that.
Okay, thanks so much. Really helpful. Thank you. Bye.
Okay. If I have another question here.
Uh-huh.
So you mentioned like use the outside marker to find ROI. So how do you like validate the ROI you found, or like how to estimate the performance of the ROI?
Super hard. Super hard. This is one of the, not weak points, but like yeah, essentially like the challenges, right? It's not easy. So in this particular case study, we were lucky because people have looked into bone and cartilage using other technologies.
Mhm.
And there were some molecules known to be specific to cartilage and molecules known to be specific to background. And so we could figure out their masses, we could figure out where they would be in the spectra. So essentially from the literature. I think a better or more kind of general way of doing this would be to have a pilot study,
Mhm.
which looks specifically, before you even can attempt to figure out any biology, just saying, okay, all I want to do is to find the cartilage, right? And work on that, figure out what the markers are, make sure that your workflow can detect that reliably, and then you go into the biological question. And so then you will not have the double dipping. And then the third option is alternative modalities, right? So maybe you can use indeed the H&E to find your region of interest, right? Or maybe transcriptomics. But then in that case, you need to do the co-registration.
I
So
see.
essentially, there is no one size fits all, but there are options.
Okay.
But in terms of knowing what is best and whether we did a good job, that's really hard.
Okay. I see. I think there's no further questions. Okay, let's thank our speaker again. Thank you. Thank you so much. Yeah.
Thank you very much. It was a pleasure.
Yeah.
Thank
Great.
you.